Welcome to the Dementia Researcher podcast, brought to you by University College London and the NIHR in association with Alzheimer's Research UK, Alzheimer's Society, Race Against Dementia and the Alzheimer's Association, supporting early career dementia researchers across the world.
Welcome to the Dementia Researcher podcast. I'm Dr. Amanda Heslegrave and I'm a senior researcher at University College London. It's my pleasure to be guest hosting this week's show. Normally I can be found at the UK Dementia Research Institute biomarker factory, and I work out how best to accurately identify dementia biomarkers from blood samples. But today I'll be doing something a little different. I'm going to be joined by two people who've got new and clever ways to look at the challenge of biomarkers for Alzheimer's disease. And they're both probably trying to put me out of work, although I think that's not going to happen. My wonderful guests have got some high-tech, amazing new approaches to diagnosing Alzheimer's disease and other dementias. So in this show, we're going to discuss these approaches, their work, and how they might be applied. So let's meet the guests. And I'm delighted to welcome Dr. Catherine Bornbaum and Dr. George Stothart. Did I say that correctly, George?
You did.
Oh, perfect. Okay, so let's do some introductions. And Catherine, would you like to go first, please? Just tell us a bit about yourself.
Sure, thank you. So I'm really fortunate to serve as the head of clinical operations and partnerships at a company called Retispec. So the company is located or headquartered in Toronto, Canada. And the company is an AI medical imaging company where we're developing algorithms for the detection of neurodegenerative diseases, specifically Alzheimer's, based on a very simple eye exam that leverages existing imaging infrastructure in eye care settings. And so the focus of my work is on the clinical validation of this technology, where we compare it to clinical gold standards. So things like PET scans or lumbar puncture, cerebrospinal fluid analysis.
Okay, thank you very much for that. And then George, can I come to you?
Yeah, hi, I'm George Stothart. I'm a cognitive neuroscientist. I work at the University of Bath, and my research is on developing functional biomarkers of dementia. So primarily using EEG to try and improve the sensitivity of our measures of brain function. And so I've been working on a technique known as Fastball for a few years now, which is a passive and implicit measure of cognitive function. Yeah.
Okay. No, that's perfect because we're going to come on to that in a bit more detail very soon. But first of all, I guess we've been talking about biomarkers. I need to kind of set the scene really for why this is important. Now, in previous times, even now, if people have problems or worries, suspicions that they might be getting some kind of dementia or Alzheimer's disease, normally they'll go and see their GP, or perhaps they'll try and ignore it, because we all know that actually there are still currently no really tried and tested cures for the disease. So there's always been a bit of a stigma with being diagnosed with a dementia, and people are quite worried about that. But so the person finally decides they're going to go to their GP, and then they will get hopefully passed on to a secondary kind of medical care, like a neurologist or a psychiatrist, for further tests. And these tests, they might be brain scans, they might be cognitive testing, or they could be something like a lumbar puncture. We do use lumbar punctures to look for biomarkers of Alzheimer's disease. But this very much, I think, depends probably on where you live and the access to healthcare. It's not always equitable. But also, the diagnosis isn't that accurate. It doesn't, for example, diagnose between different types of dementia. That's not something that's readily done or easily done. And also, by the time someone actually goes to their care provider with symptoms of a neurodegenerative disease such as a dementia, they've already got that disease, and it's not likely that you'll be able to have much effect on its progression. So what we need are different biomarkers, earlier biomarkers, and more accurate biomarkers able to differentiate between the different dementias. I think that that's really important. And so we need lots of people coming at this from many different angles. And I think that a small example of that is what we've got here today. And so I think now we'll move to Catherine, who's going to tell us a bit more in detail about your research and how this fits in.
Sure, thank you. So the work that's being done by Retispec to bring this type of technology to the market, and by that I mean our retinal scan for detection of early Alzheimer's disease, is actually technology that has a deep history of development that started more than a decade ago at the Center for Drug Design at the University of Minnesota. And so the researchers there, Dr. Robert Vince and Swati Moore, developed an earlier version of this retinal hyperspectral imaging, which is what we use. And this was to measure early changes in soluble amyloid for an amyloid target therapeutic that they were actually trying to develop. So they were trying to see if there were ways that they could measure the change of the therapeutic, and they invented this approach for diagnostic purposes, sort of out of necessity for evaluating their therapeutic. And so at Retispec, we licensed that initial technology and have gone on to further develop it and incorporate the machine learning and artificial intelligence methodologies as well. And so what we essentially do is combine retinal imaging with novel tissue spectroscopy methods in order to identify the optical signature of Alzheimer's disease. And these are the specific changes that are measurable through the eye. And so because the eye can be imaged at micron-level resolution, our hyperspectral imaging enables measurement of many biophysical, biochemical properties, such as specific proteins, so things like amyloid and tau. We can see signatures of cell degeneration, microvascular changes, and other spectral and spatial biomarkers of the eye that we can see in individuals who have been confirmed to have the pathologic signs of Alzheimer's disease. So we compare these against clinical gold standards of amyloid PET or CSF, or cerebral spinal fluid. And we use these gold standards to ensure that when we're validating this technology, that we can have a high degree of confidence that what we're detecting is sound. And what we do through this analysis is we really take these incredibly rich images and we apply our machine learning and AI-driven techniques to classify the raw data that we capture into broad categories. And so these are then used to perform the detection of the specific retinal biomarkers. So in essence, from a patient perspective, you come into an eye clinic or a clinic where this, it's called a fundus camera is. You sit down, you have a couple images of your eye taken similar to what you would have in your eye doctor's office. But what's different is that we have software that's working in the back end to capture this additional data. We replace the normal sensor in a fundus camera. So normally you would have 3 color channels, red, green, and blue. And we capture those plus over 100 additional channels. So the patient experience isn't any different, but we capture this extra information and apply the software that allows us to compute a score and to indicate whether or not that person is likely to have the same sort of protein composition as someone who has Alzheimer's disease. And the reason we're able to do this is because the retina, which is the back of the eye, is directly connected to the central nervous system and shares nervous tissues and vasculature with the brain. So it's a really wonderful way to be able to understand what's happening in the brain in a very non-invasive and simple way.
Okay, so that gives me a question. The first one would be, is this something that is already in you call it eye doctor offices, I would say opticians, but would it be something that they already have, like today?
So that's a great question. So the cameras that we use, the fundus cameras, yes, absolutely. What's different about what we do is there's a different sensor, so the hyperspectral sensor. So we work with a wonderful company called Topcon, and they have a very, very significant footprint in optician clinics, eye doctor clinics. Here in Canada, we
Yeah.
have optometrists and ophthalmologists.
Okay.
And so we're able to leverage that existing footprint.
Okay.
So doctors don't necessarily have to buy a whole new camera. We just add on our sensor and software.
Okay. And then does this technology allow you to pick up an Alzheimer's signature
Yes,
before, early, before anything else? Or would you suddenly pick up this on someone and then have to tell them?
there's great questions there. So I'll answer the first one first, and then we can chat about disclosure in just a moment. So relative to the early detection, yes. So we have completed some work just very recently that was supported by the Alzheimer's Drug Discovery Foundation. And we were looking at whether or not we could detect the signature of the proteins of Alzheimer's disease were specific to amyloid prior to symptom onset. And we, both in sort of clinical validation and in tissue samples of matched retina and brain tissues from those sort of throughout the continuum of the disease. And I'm very pleased to say that, yes, we were able to detect it before symptom onset in both instances. So it does have very promising capabilities for future screening purposes on a wide scale. And you asked a very important question also about disclosure and sort of what does this mean if we're detecting this in eye care settings where folks don't normally have sort of complex brain health discussions. And so that's something that we're exploring right now through another project that's supported by the Davos Alzheimer's Collaborative, where we actually have a system deployed here in Toronto at an optometry clinic. And essentially folks who are 65 and older, which is the age here in Canada where routine screening is recommended and covered by the government. When folks come in, they're asked an extra screening question during the history taking process about whether they've noticed significant changes in their memory. And if so, then they're invited to have a scan. So we're actually testing this in the real world in eye care settings. For now, we're not providing results. We're not having the optometrist provide the results, but we are referring to primary care for that discussion. And so we're not trying to completely disrupt, but we are using it as a way to facilitate the measurement. And then the conversations happen with someone's general practitioner afterwards.
I suppose when you ask that question, this is just me and a bugbear I have about significant memory problems. That's too late, isn't it? In my opinion, that is already too late. And so, but that's, neither here nor there. That's what makes, I think, the neurodegenerative diseases so difficult, is that they're there and then you can't do anything about them. So I guess what will make it easier is when drugs like lecanemab are proved to be useful and safe, and you can take them from a younger age. That would be the key. Sorry, I realise I've hijacked that with my ideas.
No.
And I feel like we need to bring George in now so he can explain a bit about his technology and how that made things different.
Can I just ask Catherine a question first? So if amyloid, if there's a sort of cascade or a definite process of amyloid accumulation through the brain, it doesn't arrive equally across the brain on any particular day, right? We have areas of the brain that are known to aggregate amyloid early. Where does the retina sit in that kind of process?
It's a great question. And so what we're seeing, at least from the results of the work that was supported by the Alzheimer's Drug Discovery Foundation, is that we see an increase in the soluble amyloid sort of up to I'll call it sort of intermediate stage of what we would see in brain amyloid aggregation. So it's certainly in the earlier to mid phases of that buildup. And that also is likely because we're looking into the soluble versus the plaques.
Right.
Yeah.
Okay. Is it as good at detecting tau and other markers as well, or is it kind of better at picking up amyloid?
It's a great question. So we don't have the results public yet, but our team has been working very, very diligently on both total tau and phosphorylated tau comparators. And while I can't share the sensitivity and specificity, though I do know them, they're very, very promising. So I will say amyloid is certainly not a catch-all. It's not the only measure we need. What we're building towards is even beyond amyloid and tau, looking at things like TDP and other types of markers that we know are important in neurodegenerative diseases. And we're, looking to really develop a single user experience where we can provide a profile of scores for each of these markers. So certainly not limited to amyloid, and the hyperspectral retinal imaging is really quite a powerful tool for elucidating optical signatures of various proteins and underlying biological processes within the eye, which give us an indication of what's happening in the brain.
Yeah, very cool.
That could be amazing for differentiation if that could work for the other things like TDP. That could be.
That's our goal. So far the evidence is indicating that we may have some promising options here. We're starting with amyloid, but that's certainly what we're looking towards in the future.
Okay, so now George, you can't get away with it any longer. You have to tell us research.
Yeah, happily. That's one thing researchers are always very good at, right? Talking about their own research. So as I said at the top, I'm a psychologist. Within psychology, I specialize in cognitive neuroscience side of psychology, and I spent my PhD using EEG to try and look at non-memory related signatures of dementia. So my supervisor at the time was looking at vision and visual attention, and so I was trained to use EEG in sort of traditional ways. Things like event-related potentials. These are measures that we can take, and they reflect kind of the brain's responses to particular stimuli and events in time. And I spent 4 or 5 years testing dementia patients with these techniques to try and see whether there was anything more sensitive by using whether EEG could be a more sensitive marker of certain cognitive deficits than, and it's always we're comparing to something else, more sensitive than pen and paper tests. So our traditional neuropsych measures of cognitive function. I learned a lot and hit a lot of barriers. And by the time I finished my PhD, I couldn't really see that I couldn't see the way in which I'd been trained to use EEG ever translating through to being a clinical tool. The sensitivity of these measures was never there. And while you could learn interesting things about groups of patients versus controls, for example, the reliability of these measures at an individual subject level were always terrible. You always had to record for up to an hour worth of stimulation. You then had to go through lots of reductive averaging processes and so on. So there's lots in the signal processing that just didn't really add up. I'm not a great kind of theorist. I'm not smart enough to be that type of academic. I always wanted to translate what more intelligent people than me had discovered into viable clinical tools. And that's definitely what I got a sense of in my PhD. I saw this huge gap between the way we used EEG in the lab and the way it was being used in hospitals. So the way EEG is used diagnostically doesn't look an awful lot different from the 1970s, I don't think, in most neurology clinics at the moment. There is this massive kind of chasm between experimental EEG and clinical EEG. So I saw that as a gap, and I finished my PhD thinking, well, there's all these barriers to conventional approaches, and so I spent a few years as a postdoc trying different ways, different techniques. And then in 2015, I went to a conference in Leipzig, and a very, end of the day, poorly attended poster session, I came across this poster by an academic, a French or Belgian academic called Bruno Rossillon, and he was presenting this technique called fast periodic visual stimulation. And it was a way of using EEG, and specifically a way of presenting stimuli with EEG, that seemed to solve all of the problems that I'd been hitting in the lab. And what these fast periodic visual stimulation experiments they had done and shown was that if you presented stimuli in a very specific way, so fixed periodic rates of flashing images of stimuli, and you embedded rare novel images or different images within that stimulus train and recorded EEG at the same time. Using that technique and working purely in the frequency domain as opposed to the time domain, so it's a technical difference in the way you're using the EEG data, but it made a fundamental difference to how quickly you could get reliable measures. He was claiming that using this technique, you could get significantly stable responses from individual subjects in minutes, in completely passive tasks that reflected, in his case, it was reflecting face processing. That was the particular cognitive function that he was working on. So to me, as an experimental kind of EEG person, this was a method that could give us a measure of how well a brain was doing a task, but it could do it quickly, it could do it passively, and it could do it with all the signal-to-noise ratio benefits that you would need if you were ever going to use something like that in clinic. So that really kind of caught my attention. But at the time, he had about 2 or 3 papers, and they were all on face processing. Now, face processing isn't really a cognitive function that drops off too badly in dementia. So it's a neat tool, but it's not right for dementia. That's not a cognitive function we need to measure in the early stages. So I came back to the UK from that conference and thought, can I adapt this to measure any cognitive function that might be useful for dementia? And so that's what I've spent since 2015, that's what I've been doing. So I started small, I started with memory, semantic memory, and I've built up over time a battery of tests that all use the same approach, but to measure different cognitive functions. And my ultimate sort of end goal is an equivalent battery that covers the same bases, cognitive bases, as something like the mini mental state exam or the Adam Brooks cognitive exam, these general catch-all cognitive neuropsych exams, but that we could do in minutes and passively. And so the most success I've had, or the thing that's probably the most developed, is a recognition memory version of this task called Fastball. And the way Fastball works is you see a few images, up to 8 images presented on screen. You don't do anything, you simply watch the images, and then they are embedded in an image stream that subsequently appears, where lots of images flash up very quickly on screen. Nearly all of them you haven't seen before, but occasionally these previously seen images pop up. And hopefully, if your medial temporal lobe is working as it should, you'll get a little recognition flash to those images. Your brain will implicitly, quickly go, I've seen that before. And it's that function that drops out in early Alzheimer's disease. And so what we've been able to show is using that technique in 2 minutes in a completely passive task where the subject gets no task instruction, provides no response, either behaviourally or verbally. We can measure their recognition memory, and we've shown that this response drops right out in Alzheimer's disease patients, and more recently in mild cognitive impairment patients as well.
Okay.
So that's the core of it.
Can you, with that recognition one, do you think that you could define an AD and a MCI? You've got a clear enough cutoff to do that?
We can, with our current Alzheimer's disease data and there's small sample sizes.
Yeah.
So there's a heavy caveat on it. But with our pilot Alzheimer's data, comparing Alzheimer's patients versus controls, we can get a classification accuracy of 92% using that recognition memory.
Okay.
Yeah.
And how long does it take to put the head on? You know what I mean?
Depends how fancy your EEG equipment is. And so there's a really broad range these days. When I did my PhD, there wasn't. There was lab-based EEG, very expensive to buy, took a long time to put on, got you great data, but was not an easily usable tool. In the last 10 years, the more mobile, simpler systems have come through, and we're on the verge now of really wearable EEG. That's where a lot of the product development and kind of a lot of companies are moving towards is really simple, low-level, wearable EEG that you could pop in your ear or wear on a headband. So really low profile, you can wear while you sleep or wherever. So there's a real range. And if you use one of those low burn, simple headsets, it is minutes.
And so I know that you're kicking off this study in a few weeks. I know this because I'm going to come. Can we try it out when we come? Like,
yes,
have it there?
yes,
Will you
yeah.
do a test with me? Yeah.
Yeah. So what you're talking about is well, the contracts aren't signed yet, so I can't actually say who's funding it and who's involved. But that's okay. I can say something's going to happen.
Okay.
So yeah, we're on the cusp of starting a really large scale clinical validation of Fastball using exactly one of these low burden, easy to use headsets.
Okay.
Yeah.
That's great. So you've got the recognition here.
Yes.
There are going to be other aspects of cognition that you're developing tests for at the moment, I guess?
Yeah, absolutely. So it's very well set up, obviously, to measure vision and visual attention. So those are two bases that we're trying to cover quite comprehensively, because there are also a lot of use for other neurological diseases as well. So there could be opportunities to use those in other degenerative disorders. Memory, language, vision, and attention, we have versions of the task for. The only one that it's not very well suited to is anything that requires what psychologists call executive function. And so that means doing a task. So remembering instructions or providing a response or decision making, because the nature of the task means stimuli have to be presented very quickly, multiple images per second. And so you don't have the time in that to discriminate, say, between two stimuli or make a decision on any stimuli.
Okay, so this is something that would work alongside other tests, but potentially making those tests quicker and easier, because I know that cognitive testing can be quite lengthy.
Mm-hmm.
Or it might just be that the people I know who do research call them in for a whole day of tests, and it sounds
absolutely.
tiring.
Yeah, it does take a long time, and there are I don't anticipate this ever being. I think it's very unlikely that any biomarker will be the silver bullet and there will just be one. It will be a combination of the best things that make it through this, the next 10, 15 years worth of research and development. But this will, my hope for this is that it could, to some extent, replace lengthy neuropsychological testing in situations and environments where you don't have the time or resources to do it. And because it requires no comprehension of the task or response, it means it's completely independent of language, education, and culture. And that could be really useful. So if you wanted to test, in countries with less well-funded healthcare systems, EEG is cheap, and this. You could use this test equitably across populations, in theory. I haven't tested that yet, but in theory.
How easy is it to interpret the results from it? Or do you need to be I don't know, what do you need to be to interpret the
Well,
result?
you do need to be a neurophysiologist, really, to take the raw data. But what we hope to do in the project that's coming up by working with a tech company that does a similar thing to what Catherine's company do is
Okay.
put a lot of smart
Okay.
processing and AI
Yeah.
behind the tech, so
Yeah.
that you remove the need for someone like me in the room, and
Yeah.
instead you just provide a clinician or a healthcare technician with a score.
Oh, so I feel like I'm the only person here who's not using AI or about to.
Well,
I feel,
I don't know the first thing about it, but yeah, again, people cleverer than me do.
I feel I might become defunct soon,
Yeah,
except that you guys all keep asking us to come and validate your work. So I
yes, that's
think we're
true.
all right. We're okay for a while yet, I think.
Yeah,
Yeah, you're not just going to take my
yeah.
job away immediately. Yeah.
No, 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 no.
Yeah. Okay, so that was all really, really interesting, and it's good to hear about the different strands that go up to make, all of what we do good. But I suppose we should now ask just a few nice questions, such as who inspires you, or what inspired you to the work in the dementia field that you do. Catherine first.
Thank you. So on a personal level, I really appreciate a very complex problem. Just it's what drove me as a scientist back when I focused exclusively on that, and it still motivates me here. I do have some personal connections with loved ones, so I've seen what the disease can look like, and even I'm seeing people that seem younger and younger, and maybe that's just because I'm getting older and older, but the number of folks that I know and love that are impacted by this disease, both with a diagnosis and as a caregiver, it really motivates me to wake up every day very early, start very long days, and remain focused on bringing accessible, scalable, accurate, easy diagnostics to the market, and to support other champions who are working on therapeutics. So it's deeply personal for me.
Okay, thank you very much. And George?
Yeah, I think initially my inspiration were my two PhD supervisors. So I didn't come to them with a predisposition to want to work in dementia. It was my supervisor, a lady called Andrea Tales, who's now a professor in Swansea, and she was just explaining her EEG work with Alzheimer's patients, and it seemed fascinating. I was always interested in biological psychology and neurodegenerative disease, and that just seemed like a really worthwhile thing to be spending my working kind of time on. Then, and the other supervisor was a lady called Nina Kazanina, who taught me how to use EEG and was just incredibly smart. And I'm very lucky with my two supervisors; they really kind of inspired me. But then, as I spent more time in the dementia research field, it just reinforced that desire to keep working in here. And then, as time progresses, like nearly everybody, eventually someone in your family develops it. And so, yeah, I've seen close family members develop and die from different forms of dementia, and yeah, that certainly motivates you. But so does working for, kind of face to face with patients as well. The one upside to working with what can be a very devastating disease is you do see, or I certainly saw some very, very heartwarming sides of human kind of partnerships, I suppose. So whenever I would test people, they would nearly always come with their partners. And you would see, for somebody who had real short-term memory problems and would repeat themselves endlessly and get stuck in verbal kind of repetition loops, ask the same question 30 times in 5 minutes, you would see their partners demonstrate just the patience of saints, and that was kind of really, really lovely to see. You see this kind of these couples that have been together for 50, 60 years, and the patience and the tolerance they have. They never lost their temper. There was never a cross word. They always just answered the same question again, even though they've just been asked it 29 times previously.
Oh, I think sometimes actually, like I like to remember that the samples that we get are from people, because sometimes it's a step away from it, and
Yeah,
so
I'm
I
sure.
like whenever I go to a conference and there's a real life, so someone will come in who is living with dementia and
Mm.
speak. It's always a like, yeah, that's why we do what we
Yeah,
do. That is why we're here. So that's
absolutely.
really nice
Otherwise,
to
they
be.
just become acronyms, don't they? It's
Yeah,
AD and BLB and
yeah, AD, CSF, and yeah,
yes,
exactly.
yeah.
Um, Well, I just want you to just consider the ECRs, or early career researchers, who are listening to this podcast, and what advice would you give to them? Very brief piece of advice you'd go into them if they were coming into the field today. Catherine first.
Sure. That's a great question. So I think a few things perhaps. So first, to identify a problem that you feel very strongly that there's a solution there for. And by that, whether it's, a specific protein or a mechanism, or an implementation challenge, whatever your area of expertise is, to really target that problem. And also to ensure that you're working with great mentors who can advise you, give you feedback on your grant proposals, things like that. People who've been very successful in hitting the milestones that are important for an early career researcher. I think it's also really important, aside from the sort of academic or the scientific focus, to make sure that you maintain a part of your life that is separate from your work. I think it can be very all-consuming at times, especially in those early career years where you might be working towards tenure or, really ambitious aims, or, trying to carve out your first big projects that you're running independently. It is important to make some time for life in there as well, whether it be with family or friends or just something for yourself. I think a lot of early career researchers can kind of get lost in that. So, trying to target something specific that's just yours above and beyond. Hopefully that's helpful.
I know, and especially the bit about work-life balance. Honestly, some people really do need to hear that and look after themselves. And George, your advice?
Yeah, well, just to continue the work-life balance theme, I always got, especially as a PhD student, I often saw an example of sort of reinforcing attitudes that academia required total and absolute dedication. That if you weren't working weekends and evenings, you weren't doing it right. That's rubbish, and don't. And it's just not true. And don't get drawn down that path, because it's not it's not healthy, it's not productive, and it's not needed. I think if you're organised enough with your time, it shouldn't be this life-consuming career. It doesn't have to be at all. And Catherine's absolutely right to make sure that work-life balance is maintained. I think just for my field, just for ECRs in cognitive neuroscience, I think you're lucky, because I think the employment opportunities you have in 10 years' time or 5 years' time are going to be very different and broader, perhaps, than when I finished my PhD. I think the role of industry and health technology companies in employing PhD students and furthering research, I've seen that really start to kind of explode in cognitive neuroscience, and I don't think that was really there 10 years ago, and or it was just starting. And I find that exciting. I like that. I think it's good that you can potentially finish a PhD and have a range of different places to go and work, and you don't have to just stay in academia necessarily if you want an exciting career in research.
Yeah. Okay. Well, this has been a really interesting conversation, but I think we're going to start to wrap it up now. But I need to say, if anyone's watching or listening, and they feel like they would like to host a podcast on whatever area of neuroscience they want, then that's fine, or dementia that they want, that's fine, or if they want to be a guest, so please contact Dementia Researcher. Also, I'm looking here, there's also plans for a new series where mentees interview their very inspiring mentors. And so if that's something that you'd want to be involved with, have a think about who's inspired you or who continues to inspire you and who would agree, obviously, to be interviewed by you as well. That's got to happen. So yes, in the meantime, I'd like to thank both my guests, Catherine Bornbaum and George Stothart, who've been absolutely amazing, and it's been really, really fun to hear about your research, and I've really enjoyed hosting today. So thank you very much.
Yeah, cheers. Thank you, Amanda.
Thank you.
I'm Amanda Heslegrave, and you've been listening to the Dementia Research Podcast. Please remember, leave us a review and let us know what you thought about the show. Thank you.
Brought to you by dementiaresearcher.nihr.ac.uk in association with Alzheimer's Research UK, Alzheimer's Society, Race Against Dementia and the Alzheimer's Association, bringing you research, news, career tips and support.